All right, we're started. Go ahead.
Okay, so hey guys, this is just our lesson 12 group problems. So for number one, so it gives a list of terms and it asks which ones are associated with what. And so for A, it asks which ones are examples of regulatory proteins. And that's going to be the activator and the repressor. And all of these just come straight off the notes. Most of them you could just find real easy. So for B, which is an example of a small effector molecule, that's going to be allolactase and CAMP for CAMP. And for C, which is an example of a sequence found in the DNA, that's going to be the CAP and then the -10 and the +1 sites, because those two are both part of the LAC promoter. And then it's going to be LAC-Z and LAC-A. And for D, which is not involved in the LAC operon, I said the activator, the inhibitor allolactase, CAMP, the repressor, and the inducer. So for E, which is an example of a structural gene, LAC-Z and LAC-A are both structural genes. All right, so for number two, again, fill in the blanks, pretty simple, all came from the notes. The LAC operon is regulated by two inducers, allolactase and glucose. Okay, for B, the binding site for sigma factor in RNA polymerase is called LAC-P, while the CAP site is the binding site for the activator that regulates the LAC operon. And C, the enzyme adenyl cyclase converts ATP to CAMP. And D, the -35 and -10 and +1 sequences are found in the LAC-P portion of the LAC operon. E, the LAC-L gene encodes for the LAC repressor, while the LAC-Y gene encodes for lactose permease. And F, it's called attenuation, can regulate gene expression in bacteria by causing transcription to terminate prematurely. And G, so E. coli strain that contains two copies of the LAC operon is called a merozygote, which is what the three guys used in their project. So for three, let's see. So we're supposed to figure out if the cells are going to be blue or not. So for A, I said that there wasn't going to be any blue cells because if the IPTG is absent, then there's nothing there to take the repressor. So then the gene's not going to be expressed if it still has the repressor. And for B, I said no blue cells because the cell needs glucose to undergo catabolism. And for C, it's the same thing. Catabolism can't take place because there's no glucose. D, I said that the blue cells are seen in D because everything's present, everything's there, good to go. And for E, because there's no X-gal, that shows us that there's no beta-galactosidase as well. So that means that the reaction is not able to take place, so there won't be blue cells in that one either. All right, on to number 4.
All right. Number 4. Okay, so below, so we're given, let's see, A, B, C, D, E. So we're given 5 different options for the LAC operon genotypes of several E. coli strains. And we're supposed to determine, based on whether these genotypes, whether they're defective or one of them is a repressor that can bind to the operator but cannot bind to the allolactose, so it just stays on the operator. And so we're supposed to determine if they'll be constitutive, and constitutive just means that they're constantly producing, like nothing is keeping the RNA polymerase from producing the different structural genes from LAC-A and LAC-Z and LAC-Y. So that's what constitutive means. It's constantly processing it. Inducible means that it's perfectly regulated, that it's regulated like when there is a lot of lactose, like it is creating plenty of genes to, or plenty of proteins to take in that lactose. And then non-inducible means that they can't produce the proteins to take in the lactose. So that's just not happening. It's non-inducible. It's unable to do that. It's not regulated. It just doesn't happen. So for A, all of the genes are wild type, and so I said that it'd be inducible, meaning that it's regulated. There's no defects or anything, and so it's perfectly regulated. For B, we know that there is, for the I superscript S, that for that repressor, or for LAC-I, it produces a repressor that just stays on the operator. Therefore, it's non-inducible, meaning that it's never able to create proteins that take in the lactose. So it's just not able to happen because that repressor can't come off the operator. C, so this has a chromosome and a plasmid, and so has a hole. We're supposed to determine if it's inducible, non-inducible, or constitutive. So has a hole for C, it would be constitutive, meaning that it constantly is happening, because the promoter is, I'm sorry, the operator, where am I? Oh, I missed my place. Okay, the operator, this is kind of confusing to explain, but the operator, it's not able to, it's a defective operator sequence. So the repressor isn't able to bind to the operator. So it's always occurring. It's always producing proteins. So that means it's constitutive. And that's one of the plasmid that's the case. And so as a whole, it is constitutive. And then D, it's non-inducible, because for this reason, the LAC-I gene is producing an I superscript S, which means that it is always bound to the operator of one of them. And it's also able to bind to the operator of the other one as well. So it's non-inducible, meaning that it's not able to produce anything. E is constitutive. And the reason this is because OC, the operator is a defective sequence, and so, but everything else is normal. But not even the I superscript S can bind to that operator. Therefore, it's constitutive. It's constantly producing. I know that could seem kind of confusing, but hopefully that made somewhat of sense. And then 5 is actually a similar question. It just has to do specifically with the Jacob, Monod, and Pardee experiment. And so you're given a chart, and you're supposed to determine if they're put in a tube with lactose and if they're put in a tube without lactose, if they're 0, meaning that they produce no yellow color, or 1, meaning that the amount of yellow product when one copy of the lac operon is transcribed, and 2 indicates that would be both the chromosome and the F prime plasmid are producing producing 2 copies of the lac operon, if that makes sense. So for A, for that strain, in lactose, it's 0, and without lactose, it's also 0. It's not creating any, and that's because the I minus gene is defective, meaning that it's not able to create a repressor. And then in B, it creates 1 operon in lactose and 1 without lactose as well. C, with lactose, it creates 2 yellow color, if that makes sense. And that's because both of them are producing the lac operon regardless, because the operator in the plasmid is OC, which is a defective operator sequence, meaning it's constantly producing it. And then when lactose is in there, it's also constantly producing it. And so that's why there's 2. But without lactose, it's just 1, because the chromosome isn't able to produce it, because the chromosome needs lactose in order to produce those operons. D, with lactose, 1 is created, and that is due to the promoter in one of them is defective, but the promoter in the other isn't. And so the repressor created is able to go to the other promoter. And then when lactose is present, it it unbinds from that promoter and it's able to go through. But when lactose isn't present, that repressor is bound to the operator still. And so when the lactose isn't present, it's 0. E, both are 0. With lactose and without lactose, it doesn't produce anything. That's because the I superscript S, that is always bound. It can't unbind, and so you can't create any operons. So I'm sorry if that was somewhat confusing, but hopefully that made sense to some of you guys. And yeah, that concludes our video. That's all the questions. Do you have anything else to add, Haley?
Nope, that's it.
All right. Goodbye.